Hi, good evening, everyone, and welcome to this month's ACR RFS AI Journal Club. I'm delighted to be here with you tonight and help to moderate this session. My name is Dan Cohen, one of the advisory council for the Journal Club. Some brief notes before we start. We have approximately 40 minutes of a presentation that will be followed by 20 minutes of a discussion, question and answer. You are very welcome to post your questions on the webinar chat feature, and we will try to answer them in the discussion. Just in case, the lecture is recorded along with the prior presentations on this link, just in case you miss some valuable information. Please tweet are highly encouraged at RadAIJC. So any comments, thoughts about the webinar are very welcome. I also want to thank many of the advisory council members who have been helping in the organization and contributing to the Journal Club, including Lindsay, Jeff, Teresa, Cyrus, and Atif. So as for today, I'm delighted to have with us two neuroradiology fellows from UCSF, which used to be together at UPenn as residents. Both are incredibly talented and amazing people. Dr. Andreas Ross-Checkers and Dr. Jeff Rudy, which also happened to be one of the advisory council members. I won't say much, but overall I was impressed to see how they essentially replicate the way that we as radiologists came up to a differential diagnosis. So thank you everyone to be with us, and I will be passing now the screen to Dr. Ross-Checkers.
All right, thank you very much, Dan, for that introduction, and thanks to you and the ACR and to Stephanie for having us present our paper here today. So my name is Andreas Ross-Checkers. As Dan said, I was a radiology resident at Penn together with Jeff Rudy, and we'll be presenting our work that originated during our residency at Penn here today. And this was recently published in this paper that I'll be talking about in radiology with the title that you see on the screen here, as well as another paper that's currently in press at Radiology AI that Jeff will cover just after this short introduction here. There were a lot of different people involved in these projects, and both from the clinical side and research side, and I definitely want to acknowledge all their contributions. You see the many authors of the paper on the screen here, and I'll have another acknowledgement slide as well. I guess with that, I'll get started, and you can already see here on this slide, if you've been watching the movie, sort of what the main point is of what we're trying to accomplish here, which is on the left side of the screen, you see a clinical brain MRI, and we wanted to use real clinical brain MRIs as input and be able to pull out features from these images that neuroradiologists know or think to be important for differentiating between diseases. And so we do that, we pull out a number of different features, which I'll talk about. And then the output, we wanted it to be a probabilistic differential diagnosis of diseases for that particular patient. And so I'll talk about the various steps and how we accomplish that, and then how such a system might perform in relation to radiologists. So again, I want to acknowledge a number of people, both at Penn, where this work originated, as well as the UCSF, either the project itself or follow-up projects from this. Here are funding sources as well. So thanks to all of those who helped out. Now, probably many of you listening here today are clinical radiologists, and those of you who are know that the imaging volume and the complexity of imaging is increasing at a really unsustainable rate. And so many people have suggested that this is actually potentially leading to an increase in the errors made by overworked radiologists. And in fact, that article in the popular press was based on this article published actually now 5 years ago already. So things have only gotten more busy since then. But at that time, they concluded that radiologists were having to interpret one image every 3 to 4 seconds in an 8-hour workday to meet workload demands. And I would also argue that probably a lot of that time is spent not really doing those things that radiologists were really trained to do, and just, not really practicing at the top of their license. And so wouldn't it be nice to have some help to save some of that time? And then on top of that, medical knowledge is really getting more and more highly subspecialized. And so unless you're a subspecialized neuroradiologist, you may not know if you're confronted with a young woman, let's say, with memory difficulties, and this brain MRI, which of a number of different possible etiologies could account for these findings. And these different etiologies have very different implications for follow-up, for treatment, and so forth. And the current state of things is that around the world and around the US, there are not always subspecialized neuroradiologists available. And so, it's that combination of things that made us think, could we develop an AI system to address those limitations? So our approach from the beginning was to try to explicitly computationally model the 3 fundamental steps to image interpretation that radiologists use. And that would be detection of image abnormalities, and we do that using convolutional neural networks. And then characterization of those imaging findings or those image abnormalities that we see, and we do that using a lot of different types of image processing that I'll talk about. And then number 3, we integrate those image findings with some clinical characteristics or demographic characteristics to create a differential diagnosis. And we do that using Bayesian Inference. And I'll talk a little bit about that as well. So our initial proof of concept project that I'm going to be talking about was for 19 different diseases that affect the brain. And you can see FLAIR images of those 19 diseases here, and one example for each. And you can see that the FLAIR abnormalities are similar appearing between some of them. They're very different in other ways. But you can see that, and actually neuroradiologists know that different kinds of features will differentiate these diseases. And of course, that's what imaging is based on. And so some of those might be clinical characteristics. So for example, some of these diseases tend to occur in younger age groups and some in older ages. Some you might see mass effect on imaging and others you wouldn't. Some show more symmetrical lesions and others more asymmetric. And some of these types of diseases may, on average, show post-contrast enhancement, whereas others don't. And the argument that I'll make, or what this was based on, is the idea that if you know the probability that each of these diseases is associated with certain features, then if you're confronted with a new case and you're able to extract all of these different features that differentiate these diseases, you can come up with a probability for each of these diseases of for that particular case. And that's the principle of Bayesian inference. And so then the first question is, how are you able to extract these different features that you know matter? And so first we have to find the lesions in the first place. And so that's the detection of image abnormalities, which I'll talk about first. And so this takes us back to an earlier project that ended up being published in AJNR last year. And this was essentially building a UNet to be able to detect flare abnormality. And this UNet was trained on 295 cases of flare abnormality with varying pathologies from various scanners and with various acquisition parameters, and then was tested on 92 independent test cases. And so here you see some example test cases with the output of the UNet in the middle row and the manual segmentation of lesions in the bottom row. And you can see that there's a lot of variety in the shape and size and distribution of abnormal flare signal. And you can see that it seems to capture that quite well. And if you quantify that performance using a dice metric, and compare the UNet, which is in green here, to radiologist to radiologist dice, so two different radiologists compared to each other, you can see that it's very close to human performance across these different diseases. And these were just two other algorithms that can identify white matter signal abnormality and that are not deep learning based. And you can see it outperforms those. And if we look across the 19 different diseases, you can see that there's actually a lot of variation. If you just focus on the blue bar of human to human dice scores across these different diseases for various reasons, which we can talk about if people are interested. But you can see that the UNet really follows the human performance overall across these diseases. So it does quite well across a lot of different kinds of pathologies. And so, obviously one thing we get from that is just a measure of total lesion volume, which is valuable in and of itself. And you can see that the correlation between true total lesion volume and predicted total lesion volume is very high and is quite accurate. Now, again, as I mentioned, we used a lot of different acquisition parameters, both for training and then also in the test cases. And these came from different field strengths, 2D and 3D acquisitions, from different scanners and different TEs and TRs and so forth. And that I think is quite important for getting these algorithms to generalize to new settings. And what we showed, and I won't go into this in detail, but please refer to the paper if you're interested, is that essentially there was not much dependence, no real systematic dependence on any of these technical factors. In other words, the algorithm didn't perform better on some scanners than others, or certain field strengths or anything like that. And so since then, we've also built other UNets for other signal characteristics. So for GRE shown here, and for abnormal T1 signal shown here. And that's all I'll mention about that for now, just for the sake of time. And I want to get on to the second stage. So let's say we've identified abnormal lesions according to abnormal flare signal. How do we then characterize these lesions? What's important for making a diagnosis? So again, here I'll just show you an example case. So here is our two lesions in this brain MRI that were detected by the UNet. We then co-register that with other imaging modalities such as the T2 weighted sequence shown there, and we can quantify the amount of signal there. We also use our T1 weighted MRI to do automated brain segmentation, differentiating into gray matter, white matter, and CSF, as well as deep structures like the basal ganglia that Jeff will talk about some more. And then we characterize them in other ways. So for example, we detect enhancement by doing just a simple subtraction of the T1 post-contrast minus the T1 pre-contrast, and thresholding and overlaying that with our flare lesion map and seeing which lesions show enhancement. We can do something very similar with restricted or reduced diffusion, again by overlaying the DWI and ADC maps with the lesion maps and seeing which lesions show reduced diffusion. We can identify the lobes of the brain just by atlas-based registration algorithms. We can identify the hemispheres to look at things like symmetry of lesions. We also detect things like mass effect, so we automatically draw an ROI around the largest lesion that we identify and overlay that with the CSF, and essentially see if the CSF is being pushed out of the way by comparing to the contralateral side and seeing what the overlap there is with the CSF. We also measure things like ventricular volume, and this is just a reminder that all of these characteristics are identified in 3 dimensions. So even though I'm showing it for slices here, of course, everything is done in 3 dimensions. If you wanted to know which part of the brain the lesion is in, we could tell you, which Brodmann's area, or which part of the cortex is involved. We also measure distance from the ventricles to see if lesions are periventricular. We measure distance from the cortical surface to see if lesions are juxtacortical. And we measure things like how much overlap is there with structures like the corpus callosum, again, just using atlas-based registration techniques. And so we get this rich description of the lesions that tell us all kinds of features about the lesions. And in fact, we get that kind of semi-quantitative description for every single lesion in the brain. And so this makes up sort of a superset of findings, something that a radiologist might put into the findings section of a report. And so then the next question is, how do you synthesize all of that information? How do you integrate it, ideally with some clinical information as well, to create a differential diagnosis, which is inherently probabilistic? And so we do that using Bayesian inference. And so here is a sort of cartoon version of all of these different features that we extract, as well as a couple limited clinical features that we also get for every case. If we can talk about this a little bit more, but essentially Bayesian inference requires a set of conditional probabilities, a large probability table of each of these features in relation to each of the diseases that you're trying to predict. And we do that actually using expert consensus. So some neuroradiologists decide, either based on their knowledge or based on textbooks, what those conditional probabilities are, and then you can also update those probabilities by data. All right, and so for this particular example that I've been showing, if you automatically extract the features, input that into this Bayesian network, it gives you a differential diagnosis and says there's a 35% chance that this is CNS lymphoma, and that was the #1 percentage, and indeed this is a biopsy-proven case of CNS lymphoma. And so we can validate that system on a larger set of data. And so we collected cases with either clinical or pathological gold standard diagnoses. We split some of that data and used some of that as training to update some of those probabilities. We can talk a little bit more about what else this training data was used for. And then we tested the AI system as well as radiologists on 92 test cases, which is approximately 5 cases for each of those 19 diseases. And so these cases, a number of radiologists provided their top 3 differential diagnosis for those cases just by looking at the images and having some of that clinical information available. And those were radiology residents, general radiologists out in practice, neuroradiology fellows with 9 months experience, and academic neuroradiology attendings. And then we compared that to the performance of the AI system, which also gave its differential diagnosis. The radiologists saw the cases just as they would in the normal PACS that we had at Penn at that time, which is the Sectra PACS. And all cases are anonymized. They're shown all the sequences that the AI system also gets to see. And then they're given the same clinical information that the AI system gets as well. And so how do radiologists do? So these were pretty tough cases overall. Here's the performance in terms of % correct of getting the correct diagnosis within the top 3 differential diagnoses. So chance performance here would be 3 out of 19 if you just randomly picked 3 diseases out of the 19 possibilities from the dropdown. And so importantly, the differential diagnosis was chosen from a dropdown menu of those 19 different possibilities. And you can see that residents get a little bit under 60% correct within the top 3 differential diagnosis. How does that compare to other radiologists? So general radiologists actually do similarly. This black bar, by the way, is the mean across the individual subjects, which are the circles. And then neuroradiology fellows do significantly better, closer to 80% correct, and then academic neuroradiologists are nearing 90% correct. How does that compare to the AI system? That's represented by this dotted line here. It's not significantly different from academic neuroradiologists, and for at least for top 3 differential diagnosis is better than fellows, general radiologists, and residents. You can also measure performance in a number of other ways, of course. It doesn't have to be getting the diagnosis within the top 3 differential diagnosis, although we think that's valuable. So you can also say, well, did it get it in the top 2 possibilities? And there, again, there's no difference to academic neuroradiology attendings, also no difference in this case to fellows, but still better than residents and general radiologists. If you ask just, did it get the right diagnosis as its top diagnosis, sort of similar performance. Overall, the academic neuroradiologists are still a little bit better there than the AI system, but similar performance to fellows with that measure for those diseases. You know, there's a lot of interesting things about the data. One thing that we thought was interesting is that this really applies to rare diseases as well. In fact, the AI system has a relative advantage for rare diseases over radiologists, because as you can imagine, you know, a radiology resident who's never seen half of these really rare diseases isn't going to do particularly well on those. And so there's a large difference here between their performance on common diseases and rare diseases, whereas the AI system doesn't really care because it's just purely based on knowledge. You know, it can identify the flare abnormality anyway, just as a radiology resident can, and then it has sort of the knowledge within the Bayesian network to be able to identify those rare diseases. And so there's no significant difference between common diseases and rare disease performance for the AI system. And you know, as expertise increases, that gap is less and less, but it's still significant even for academic neuroradiologists. We can also analyze error rates in different ways. So you can see that the AI system sometimes confuses CNS lymphoma and high-grade gliomas. You can see that it overcalls migraine headaches, because basically non-specific flare signal abnormality can often be migraine, and it doesn't have a better way of differentiating those. And then we can look at the errors using these confusion matrices for the different radiologists as well. And basically you know, I'm not necessarily showing this, but well, the first take home, I guess, from this is that as your skill level or experience level improves, you can see that the yellow, which is the fraction, the correct, falls more along this diagonal, which means you got an overall higher proportion of cases correct. And then what's interesting, though, is that the correlation between the confusion matrices between attendings and fellows is actually more similar than between the AI system and either one. So it kind of suggests that that humans make more similar types of diagnostic errors potentially than the AI system and humans. So maybe there's something complementary there. So in summary, so far, what we've covered is our development of an automated image processing pipeline that models the radiologist's image interpretation steps. This kind of system allows for model introspection and access to all the features that eventually make up the diagnoses, because it's all of those features that we explicitly extract. And as a proof of concept for these 19 diseases that I mentioned, the diagnostic accuracy approach is that of neuroradiologists. So we'll leave questions to the end. And so with that, I'm going to hand it over to Jeff Rudy now, who's going to talk about some extensions of this work and complementary work.
All right, thank you, Andreas. great presentation. Thanks, Dan, for organizing this and the introductions, and also thanks, Stephanie, for helping organize. So I'm going to be talking about, for the most part, a sort of a parallel project to what was recently published in radiology. Let me go to the overview here. So in this system, study, we were looking at a different set of diseases that involve deep gray matter, and this is in press in Radiology Artificial Intelligence, so hopefully should be published soon. So I'm actually going to go relatively quickly through the results of this system because it's quite remarkable how similar the overall results were to what Andreas just presented for the 19 diseases of the cerebral hemispheres. And then I'm going to take some time to talk about one of the additional projects that we recently done, kind of taking the next step, asking the question of whether or not these systems could potentially augment a radiologist's performance. I'm going to take some time to talk about like big picture discussion about the benefits of this approach and limitations for the approach that we've taken here. Spend a little bit of time on future directions and conclusions, and then we'll open it up for questions. So as I mentioned, this study detailed 35 different common and rare diagnostic entities which involve deep gray matter. For this study, I actually did include normal MRIs as well. And you can see the list of the different diseases here. Similar to before, we essentially took 2 to 3 examples of each of the different diagnostic entities, as well as half of the normal cases, and sort of put that into the held out test sample and use the other cases for training. And actually, I'll mention that training of the convolutional neural networks, we actually did combine the 2 data sets. So all of the cases in Andreas's sample were actually also used for training of the convolutional neural networks. And again, multimodal brain MRIs that were collected at Penn over about a 10-year period, many of which were from outside scanners, so that a good heterogeneity in the data included. So here's a similar slide of an example of each of the different diagnostic entities. And you'll see that there's, again, a good variety of different etiologies. We have a lot of toxic metabolic diseases, infectious, neoplastic, vascular entities across these different 36 entities. And again, one of the crucial first steps of the image processing pipeline is the tissue segmentation and anatomic localization. Again, we're using the T1 images here. A pre-processing step we used was a super resolution technique as well as skull stripping. So then again, we use the ANTS pipeline to perform multi-atlas tissue segmentation to segment into deep gray, white matter, cortical gray matter, and CSF. And then, for this project, the most important anatomic regions were the different deep gray structures, caudate, putamen, globus pallidus, thalamus. So we really needed to get a good parcellation of these structures, and we actually tried a few different methods for segmentation, and like, for example, FSL, Free Surfer, and ANTS worked quite better than others, because as you can imagine, if there are lesions within the deep gray structure that change the signal abnormalities, it might disrupt where these different structures are. So you have to sort of use your spatial priors, kind of know where these regions might be. So there's a few different tuning steps we had to do to get it to work properly. And then, as before, we used different UNet convolutional neural networks for the detection of abnormal signal. Here we have example deep gray matter cases where the FLAIR UNet is detecting abnormal signal on the FLAIR images. Again, the susceptibility detection UNet, and these are example deep gray cases where there's abnormal susceptibility. And then again, a T1 UNet that was detecting abnormal signal on T1, again, noting that there was abnormal signal both on both decreased abnormal signal and increased. And then for the detection of enhancement and restricted diffusion, as described before, we used threshold-based methods with enhancement where we used both the T1 pre-image and the T1 post-image, and you used a threshold to kind of determine whether or not there was enhancement in the regions. And then same thing for restricted diffusion. And then again, here's an example for anatomic localization. We have the original T1 weighted image. We perform the tissue segmentation. Again, it identifies the deep gray structures regardless of the fact that there are signal abnormalities. And then we have the flare image, and we detect the abnormal flare signal with the UNet. And then we overlay the abnormal signal. And in this case, we used any abnormal signal, whether it was detected on T1 or susceptibility, overlaid that on top of the tissue segmentation and parcellation to determine which regions were involved. So this is a table that I think is actually pretty interesting. It's looking at the performance of each of the individual features for the image processing pipeline. So if you look at the right column with accuracy, if you look at across all the different features, this is telling you that the automated image processing pipeline was 89% accurate relative to ground truth features. And this was compared to attending radiologists evaluating each of the cases and determining whether or not these features were abnormal or whether there was increased or decreased T1 signal, etc. And then you'll note that if you look at the overall sensitivity and specificity, the specificity was on average better than sensitivity, and this was for a particular reason. To go into the details a little bit more, one of the important critical aspects of extracting these features is coming up with a sort of a threshold. That is, do you want to say that the caudate is involved if there's only a single voxel that's abnormal in it, or you have to say that at least 2% or 3% of the voxels are abnormal? So what we did was we tuned these different thresholds based on the training data in order to optimize performance overall in the training data, and optimizing performance kind of required tuning for specificity over sensitivity. You can imagine, because a false positive, if it detects enhancement when there is no enhancement, could cause downstream effects. So here's a deep gray Bayesian network. I've kind of alluded to the features included in it. Again, we have the signal features, T1, flare, restricted diffusion, susceptibility, enhancement. The only anatomic features were those deep gray structures. And then the other spatial features that were included in this network were simply whether these signal abnormalities were bilateral and or symmetric, which also were established based on thresholds, based on how much signal was abnormal left versus right. And again, we used a few different clinical features, including acuity, immune status, age, gender. And again, experts' consensus determine the conditional probabilities. And actually, I just wanted to quickly show you these probability tables, because these are included as supplementary tables in the papers, but essentially all of the knowledge about these diseases and different features are stored within these probability tables. And then for every new case, you use the naive Bayes calculation to just come up with a probability for each disease, which is actually a fairly straightforward calculation. So this was just an example case overview, again, showing the different steps of the pipeline where you can converge together and come up with a differential diagnosis. So as before, we had the same residents, fellows, and attendings review the deep gray matter cases. They actually did this at the same time as the diseases in the other study. They just alternated between the two and had a drop-down selection, reviewing the cases in packs anonymized. And so, really remarkably, for this different pipeline, different set of diseases, we had very similar results. That is where the AI system is performing at the level similar, not significantly different from academic neuroradiologists, but is significantly better than residents, general radiologists, and neuroradiology fellows for top 3 diagnosis. But then for top 1 diagnosis, similarly, the performance of the system is a little bit lower on top 1 than top 3. And you can also display this using a receiver operator characteristic, again, showing similar results across the top 1, 2, 3 diagnoses. And same as before, again, the power of this system has a lot to do with rare diseases. Again, you see that for radiology residents. No pointer here. For radiology residents and general radiologists, the performance is better for common than rare, whereas that's not so much the case for academic radiologists or attendings. And this is another interesting feature. I like this analysis because you can kind of think of it as an ablation analysis, as in what features are driving the performance in the system overall. So what we have here is basically in the test set, if you remove a specific feature, how much does your performance drop for top 1 or top 3? And you can see that Clinical features, of course, are important, and your overall performance by removing all clinical features is about 20%. Imaging, if you do remove all imaging features, your performance goes down quite a bit more at 45%. And then signal features contribute about 20%, and anatomic subregion and spatial features contribute about 10% for top 1. But again, it does vary for top 1, top 3, but it kind of gives you an idea of how critical each feature is for the overall performance of the system. And then same as before, looking at correlation matrices, there was a stronger correlation between individual attendings and fellows than there was between the automated system and attendings or fellows, again, suggesting that the automated system is making different mistakes and or getting cases correctly, somewhat different than humans. And so this kind of leads me to the second study that, actually first I wanted to just show overall, if you this image of sort of all 50 diseases, if you look at the 2 different pipelines together, we've kind of shown you a proof of concept for 50 different diseases shown here. And you can actually take the results of the different radiologist groups and combine them together. And again, the results are, when you combine them together, are remarkably similar, with AI system doing similar to the academic neuroradiology attendings and better than the other groups. So then the next question we had was, can these systems augment radiologist accuracy? And so again, now the question here is not man versus machine, but man versus man plus machine. And so to do this, we use a system that was developed at Penn known as the Adaptive Radiology Interpretation and Education System, or ARIES. And you can kind of see this animated GIF here, where you can upload a specific network, you can change the individual features, and you can see that it updates the differential diagnoses in real time. And this network actually has 2 different differentials you may notice. One is a clinical differential diagnosis, and the clinical differential diagnosis includes, the age, presentation, various clinical features, and or prior probabilities if you want to include prior probabilities, whereas the radiographic is purely just based on the imaging findings. So what we did is we recruited different radiology residents and attendings to look at the same cases that we had before, but this time what we did is half of the time they looked at the cases on their own, and the other half of the time they had the results of the AI systems on ARIES. And so here are the results for the 4 different residents. This is their performance for the cases without ARIES, and then this is their performance for the cases with ARIES, with the results of the AI system on ARIES. So you can see all 4 residents boosted their performance by having the results of the AI systems available to them, and it also allowed them to interact with the results of the system. The attendings, there was not a significant difference for them. They're already performing at the level of the automated pipeline. And so now I want to just take a step back and talk a little bit about the overall benefits of this approach and why we think it's so useful. In a lot of the literature, you may see end-to-end deep learning solutions where you collect a large number of images and you come up with a diagnosis or prognosis. We're kind of adding an additional middle step here. Instead, we're using deep learning as well as advanced image processing methods to come up with these intermediate quantitative features. And for these studies in particular, we're looking at a secondary model of diagnosis, differential diagnosis, but you can take these intermediate quantitative features and do other things with them. You can look at prognosis potentially, you can look at longitudinal assessment of change, and then they can also be used to develop large databases for scientific discovery. If you can imagine, all the cases are run through this pipeline, you have a very large database of different lesions and patient information that could lead to discoveries. And these intermediate quantitative features really allow for explainability of the system. There's less of a black box. There's a lot of a push for more explainability in artificial intelligence algorithms. And again, the heart of the diagnosis model is the Bayesian network, and that's very explainable why it's choosing what diagnosis. And then the segmentation is very explainable too. It tells you what lesions it's detecting or not, and you can see those and see if it makes a mistake. But then the big picture of why this approach we think is much better for diagnosis is that by incorporating expert knowledge about all these diseases, it allows you to interpret the full gamut of common and rare diseases. And this has been discussed as sort of the long tail in medicine and radiology. There's so many different rare diseases that really an end-to-end deep learning solution is not going to work for many of these rare diseases, because if you only have a few examples of those diseases, how is a deep learning system really going to be able to come up with a diagnosis? So this approach is sort of utilizing the expert knowledge. There are multiple limitations, and the study's not perfect. So I wanted to cover a few of those and then kind of talk about some future directions, how to address those. So again, these two studies focused on 50 specific diseases. There are more than 50. The image processing pipeline itself, really focused on interaxial diseases. We're not really doing anything outside of the brain. That's not really part of the image processing pipeline. There's these other selection criteria. We did exclude cases that had multiple different disease processes at once, which is a reality, and so there are ways we could think about addressing that. We do require the T1 images for segmentation. And then this ANTS pipeline that I mentioned that we've been using does require a QC step and can take 8 hours to run the whole, the pipeline from beginning to end. I'm also going to talk about how we kind of get around that. Another big thing that could potentially be an issue is that for these studies we set the prior probabilities, that is how likely this disease is just a priori. If it was a rare disease, it's probably pretty rare that you're going to be seeing it if something's non-specific, common things being more common. So we set all the probabilities equivalent, but in clinical practice, rare diseases are rare. So one way of getting at that is, as I showed you before with Aries, you could have potentially 2 different differential diagnoses to think about the zebras, but also think about what's probably more realistic. And then a few future directions. We are, working on the next version of the pipeline, which is sort of an overlay of it here, but we're basically trying to combine the 2 different pipelines, expanding it to 75 diseases, incorporating additional signal, anatomic, and spatial patterns, and kind of improving some of the features that didn't work as well. But the whole system is very modular, and it allows you to iterate and improve. And then actually the newest version of the pipeline has the potential to run a single subject within 5 minutes, which would be clinically useful. Some of the steps, using ANTS for tissue segmentation, we can replace with other UNets. We can use other pre-processing steps called stripping that are also utilized UNets in deep learning to speed everything up. And then I mentioned the issue of multiple diseases. We've thought about this. This is a really challenging problem, but you can think of how do we as humans, radiologists, deal with multiple diseases, and generally we will quickly see something, and then for example, there's white matter hyperintensity as an old infarct, we kind of just discard it and then move on to what else is in there. And I recently published a paper that looked at the BRATS data set, which is a large database of brain tumor patients, and that does sort of a multi-class tissue segmentation, and I trained it to actually detect the white matter hyperintensities as a different class. So you could think about adding additional pre-processing steps to identify common comorbid abnormalities and kind of pull that out separately. And then, ideally, in addition to these the findings that we have here of the performance of the system to potentially improve accuracy, we also potentially would like the system to improve efficiency of report generation. The idea being that you could sort of create a fellow in a box, as has been coined before. You could think of the findings with the lesion analysis kind of populated by the image processing pipeline, and then the impression populated by the Bayesian network. So Andreas and I have showed you sort of a proof of concept AI systems that model radiologist interpretation steps, performing as well as academic neuroradiologists, and which we also found that augment the performance of radiology residents. We have a lot of future plans to expand and improve the pipeline and ultimately integrate into the clinical workflow to improve accuracy and efficiency. And again, this is a modular and iterative thing. It's going to probably require multiple rounds of improvement, titration, improving small things at a time, because there's a lot of potential issues that downstream could affect the overall results. And so once it potentially would be at the level that it could be clinically useful in real practice, it would probably require more additional steps. But I think that overall this approach is really a fairly realistic path forward for automated diagnosis in radiology, given how explainable it is, and again, dealing with the issue of long tail of rare diseases. So yeah, as Andreas mentioned before, there's a lot of people that were involved in this project, mostly at Penn in the Pixel lab that helped quite a bit. And I think now we'll go ahead and open it up to questions.
Well, I'm waiting to see if there's any questions from the audience. In the meanwhile, I would be happy if you can. So first of all, like that's like an amazing presentation. Like I think it's actually really remarkable. Like I think many people are aware of the UNet concept for detection and segmentation of anomalies, but I'm not familiar too much with like the Bayesian networks, and if you can just explain more about how it works and like how it assists with probabilities and etc.
Let me go back to the slide I have of those probability tables, because it comes down to the probability tables. There are different types of Bayesian networks, but we were using basically just a simple naive Bayes network. So the way that the simple naive Bayes network works is you have a new case, you get the features for all the cases, and you basically take that case, and for every possible diagnosis, you multiply the probability. So is it a male or female? In this case, it doesn't really matter, 50-50. You know, if it's a male, you multiply by 0.5. If the patient's old, then you multiply 0.7. So you do that. So you have a new case, you have all the features, you multiply that for all the different possible diagnoses, and then you have a value for each diagnosis. And then you would just kind of normalize it so that the percentages get equal up to a total of 100. And that's your probabilistic differential diagnosis. So it really, the power is just the knowledge in these probability tables.
And we've I'll just add to that, we've done a little bit of work looking at sort of how these conditional probabilities, so the numbers within these tables, how those can and should be set. And it turns out that a single radiologist can set all of those numbers just based on their knowledge and experience and looking things up in textbooks and so forth. And that is pretty good in and of itself. If you get multiple experts together, you end up doing a little bit better. And then you can also actually set those probabilities with data. So you can look across, let's say in my institution, what percentage of cases that ended up being, let's say, adrenoleukodystrophy, some rare disease, what percentage of cases showed enhancement, right? And so then you can set the probability using that. And so these can be sort of knowledge-driven or data-driven or some combination. And if you think about it, the knowledge in textbooks, those probabilities, you'll find some of those probabilities in textbooks, and those are essentially sort of summarizations of the data that people have seen across a long term. And so it just encapsulates that knowledge in the form of probabilities. One limitation to note about that is that, as Jeff mentioned, we use naive Bayesian models, and those actually technically assume that the different features are independent of each other, which of course they're not. So let's say T2 and T1 signal are not necessarily completely independent of each other across cases, and so so it's an assumption that probably doesn't bear out, but it turns out that it doesn't matter so much. Now, if you wanted exact probabilities that are like really true probabilities, you'd probably have to be a little bit more accurate in your modeling, as well as in those conditional probabilities. But it turns out for getting the right diagnosis, or even the top 3 diagnosis, you don't have to be, perfect. And radiologists obviously aren't perfect probability calculators either.
I see. Thank you. Like, we're slowly starting to get more questions, and I think we actually answered one of them, which was about how much of those Bayesian tables are derived from expert knowledge versus tuned from the training data. The 2 other questions are about, is ARIS a web-based platform to input and compare the calculations derived from the Bayesian conditional probability tables? So I hope that I'm understanding the question right, but I think the question is asking if ARIS can be used online, and it's a web-based platform that everyone can use. And the second,
I'll answer about ARIS. So the system that we use was just internal to Penn. However, one of the developers that helped develop it has recently developed an open source version of it, and I can share that. It's related to the results that I showed before, but it is available on GitHub, and the 2 networks that we have are available there. I can try to pull that up actually.
I see, and that's actually maybe a bridge to the last question that I have is, how does one go about starting to make such a Bayesian table for one of his favorite disease? So I think people are asking like how to start doing their own tables.
Yeah, that's a really great question. And that's basically how this project started. You just have to sort of think of the features. I mean, in some sense, it's like traditional feature engineering, right? You think of the features that you as a radiologist think matter for the diseases you're trying to differentiate. And you can brainstorm a number of different features, and then you have to basically ask yourself or ask the expert, which might be a neuroradiologist in our case, basically ask them a number of questions. So how commonly do you see enhancement in each of these diseases? What's the percentage? And how commonly do you see mass effect with each of these? And you literally just give that percentage. And then ideally, if you have some data available about that, you would use that. And so, as I mentioned, some of those percentages you can look up in textbooks and are published in papers, and some you might be able to gather from your own data, just as you would for any sort of machine learning project. So that's essentially the way to get started. So your columns could be on the it could be the features, and your rows are the diseases you're trying to differentiate, and then you just fill in the probabilities.
And I just pulled up the ARIS webpage here, and I put it into the chat as well.
Great, thank you. Now, I do have a question out of curiosity. Like, I'm just thinking, like, now, essentially you identified 19 diseases. One of them, for example, was PML, and let's say it gets complicated and you have PML iris which show enhancement. Like, how complicated can it get? I don't know if it makes sense, my question.
Yeah, no, that's a...
You can go ahead, Andres.
Thanks. Yeah, it's a good point. There's a couple of things to unpack there. One is, I think the central point of that question is, how much specificity is there in the diagnosis? And I think that's actually one of the central limitations of this kind of work, or really almost all deep learning work as well, which is that your outputs are sort of limited to whatever you give it. And so in our case, it's 19 different, in the first study, it's 19 different diseases, and your outputs will be, a probability ranked differential diagnosis of those 19 different diseases. But in reality, radiologists answer different kinds of questions and just give a differential diagnosis of 19 different diseases. So instead, you might say, is this infectious or neoplastic? And so that's a different level of specificity than particular diseases. Or you might get more specific and say, could this be PML, PML iris? and how likely is it that that it's not HIV encephalopathy or something. So there's different questions that might get asked, and obviously a system like this, or any system I know of, doesn't really address that at all.
But at the same time, you could also add an additional entity, PML with iris, and you might update different probabilities based on clinical status and different probabilities based on there being more likely to be enhancement. But it can come down to, well, how many total entities are there in general, which is probably more like 1,000. So you start to, but as you add more and more very rare diseases, you're getting somewhat diminished returns, but you can always add more.
So, yeah.
That's right.
There was a question, Dan, that I think we skipped over briefly. Maybe Andreas actually would be better to answer. It was about how much is of updating the probabilities versus expert versus based on the training data. And again, Andreas did more of that. I didn't really update my probabilities too much based on the training data. It was pretty much all just multiple experts.
Yeah, it's a good question. I mean, in general, the answer is very little. We largely based it on expert consensus, just because we didn't have that much sort of quote unquote training data available. So for the 19 diseases of the cerebral hemispheres, there were somewhere between 0 and 10 training cases available. So for really rare diseases, we only had a handful of cases, and so we put those all sort of into the test bin. And so because you don't actually need training cases to make those probabilities, and so in that case, they were not at all updated by training data. Where we had some training data, it was a combination. So basically each additional training case adds a small amount of weighting to that probability, and you can decide how much to weight it depending on how much trust you have in the original conditional probability estimate. So if it's based on a textbook and it's thought to be fact, then you might not want to update that probability at all, because it's sort of known to be that probability. But if it's a random guess by a couple of experts and there's really no data to back it up, then it's good to use more training data if possible. So I don't have an exact answer for you because it varies a little bit from feature to feature.
I see. No, but I personally, and correct me if my conclusion is wrong, but it seems that we can actually learn now, like by collecting more data and with Bayesian interference, knowing what is the most important features and less, and actually learn from the computer what is true or not, I guess.
Yeah, I think the data that I showed that where the AI system was augmenting the radiology residents is a good example of that. I showed you the results there, but also even just talking to the residents that you did the experiment afterwards, they were very impressed with the system, and they thought that they were learning based on it, because some of the diseases, for example, that they didn't know about, there actually what is an additional feature I didn't show you in areas, but you can actually take a specific disease and it'll pull up the most likely probabilities for each of them. So you can actually use ARIES and this AI system to help teach and train, and could potentially be a component of radiology education.
That's actually impressive. It's like 9:59, at least here on the East Coast, so just to respect the time of everyone, I don't see any more questions from the audience. So I think we're going to end up the session. Jeff, Andreas, Stephanie, thank you so much for the organization. I think it was personally like a remarkable presentation. And do you have like any last notes before we end up?
No, thanks so much for having us and giving us the opportunity to present this work.
Yeah,
sure.
Absolutely. Thank you again for having us.
Good. Thank you. All right. Bye, guys.
Bye-bye.
Bye.